Welcome to edupediaworld.com. I am Somja J. Nair, your online tutor for Plus 2 Zoology. In this video, I am going to talk about gametogenesis in human reproduction. Gametogenesis. The primary sex organs, the testes in the males and the ovaries in the females, produce gametes, that is sperm and ovum respectively, by the process called gametogenesis. The process of formation of the male gamete is called spermatogenesis, and the process of formation of a mature female gamete is called oogenesis. What is spermatogenesis? In testes, the immature male cells called Spermatogonia produces sperms by spermatogenesis that begins at puberty. The Spermatogonia, singular form Spermatogonium, present on the inside of wall of seminiferous tubules multiply by mitotic division and increase in numbers. Each Spermatogonium is diploid and contain 46 chromosomes. Some of the Spermatogonia, called primary spermatocytes, periodically undergo meiosis. A primary spermatocyte completes the first mitotic division, that is called reduction division, leading to the formation of two equal haploid cells called secondary spermatocytes, which have only 23 chromosomes each. The secondary spermatocytes undergo the second mitotic division to produce four equal haploid spermatids. The spermatids are transformed into spermatozoa, called sperms, by the process called Spermiogenesis. After Spermiogenesis, sperm heads become embedded in the Sertoli cells and are finally released from seminiferous tubule by the process called Spermiatia. Hormonal regulation of spermatogenesis. Spermatogenesis starts at the age of puberty due to significant increase in the secretion of gonadotropin releasing hormone. It's a hypothalamic hormone. The increased levels of gonadotropin releasing hormone then acts at the anterior pituitary gland and stimulates the secretion of two gonadotropins, luteinizing hormone called LH and follicle stimulating hormone called FSH. LH act at Leydig cells and stimulate synthesis and secretion of androgens. Androgens in turn stimulate the process of spermatogenesis. FSH act on Sertoli cells and stimulate the secretion of some factors which help in the process of spermiogenesis. Structure of a sperm. It is a microscopic structure composed of a head, neck, a middle piece and a tail. A plasma membrane envelops the whole body of sperm. The sperm head contains an elongated haploid nucleus, anterior portion of which is covered by a cap-like structure called acrosome. The acrosome is filled with enzymes that help the fertilization of ovum. The middle piece possesses numerous mitochondria which produce energy for the movement of tail that facilitates sperm motility, essential for fertilization. The human male ejaculates about 200 to 300 million sperms during a coitus, of which for normal fertility at least 60% sperms must have normal shape and size, and for at least 40% of them must some vigorous motility. Sperms released from seminiferous tubules are transported by the accessory ducts. Secretion of epididymis, vasa deferens, seminal vesicle and prostate are essential for the maturation and motility of sperms. The seminal plasma along with the sperm constitute the semen. The functions of male accessory ducts and glands are maintained by testicular hormones called androgen. Here you can see a pictorial representation of a sperm. What is oogenesis? The process of formation of mature female gamete is called oogenesis, which is markedly different from spermatogenesis. Oogenesis is initiated during the embryonic development stage when a couple of million gamete mother cells called oogonia are formed within each fetal ovary. No more oogonia are formed and added after birth. These cells start division and enter into prophase 1 of mitotic division and get temporarily arrested at that stage called primary oocytes. Each primary oocytes then gets surrounded by a layer of granulosa cells and then called the primary follicle. A large number of these follicles degenerate during the phase from birth to the puberty of a girl. Therefore, at puberty, only 60,000 to 80,000 primary follicles are left in each ovary. The primary follicles get surrounded by more layers of granulosa cells and a new theca, and it is called as secondary follicles. The secondary follicle soon transforms into a tertiary follicle, which is characterized by a fluid-filled cavity called andra. The theca layer is organized into an inner theca interna and an outer theca externa. It is important to draw your attention that it is at this stage the primary oocyte within the tertiary follicle grows in size and completes its first mitotic division. It is an unequal division resulting in the formation of large haploid secondary oocyte and a tiny first polar body is produced. The secondary oocyte retains the bulk of the nutrient-rich cytoplasm of the primary oocyte. The tertiary follicle further changes into the mature follicle or Graafian follicle. The secondary oocyte form a new membrane called zona pellucida surrounding it. The Graafian follicle now ruptures to release the secondary oocyte or ovum from the ovary by the process called ovulation. Thank you.